Okay, we're gonna talk about a couple of topics here. They are somewhat disconnected to some extent to finish off chapter 15. One of the first, okay, thank you. One of the first things that we'll talk about here right now is something called non-disjunction of chromosomes. Non-disjunction. So you have to remember what happens normally in meiosis before we talk about something where it's broken. So let's look at what normally happens in meiosis using this smaller length chromosome, the shorter chromosome, compared to the longer chromosome where it goes wrong. I need you to look on the screen, please. This is meiosis. It's meiosis 1, meiosis 2, producing 4 haploids. Same thing with this one. The event with respect to the shorter chromosome is normal, meaning you have disjunction. This is when I did this. The two homologous chromosomes meet each other in prophase 1. It's called synapsis. They do disjunction in anaphase 1, and then they do it again in anaphase 2. Do you remember all that? Yes or no? So, I'm showing you the normal event with the shorter chromosome. But then the longer chromosome is showing you what would happen if this happens. The two homologous chromosomes join with each other at the synaptonemal complex during prophase 1. They go to do disjunction and they say, no, we're going together. Obviously, this is something that is not what you want. This is when your cells start aging and you also are aging in the same process. The machinery starts breaking down. You know, if you buy a new car and you drive it off the lot, you expect to get home with it because it's just brand new, everything works. But as the car ages, now you're expecting it to start breaking down on you. Well, we're no different to some extent. Like when you're young, this is happening perfectly, everything is working. But as we age, things start getting slower and things start saying, no, we're going to go together. That is because the machinery is breaking down. The cutoff age for women is 35 years of age. So what happens is at the age of 35 and you get pregnant, let's say you get pregnant at the age of 38, the doctor is going to actually recommend that you genetically test your baby because of the likelihood of one of these happening. Now that's not going to hurt you, that's going to hurt your baby. So the cutoff age for females is 35. So some men in the room say, whew, thank God, that doesn't happen to me. Yes, it does. Non-disjunction happens during the formation of eggs. Non-disjunction also happens during the formation of sperm. But why does the doctor worry about the age of the female and not the age of the male? Because think about it. There you go. Nice. If the egg that is ovulated is defective, the baby 100% sure the baby will be defective. But men have numbers on their side. If 90% of the sperm are fine and 10% are bad, what's the chance of you having a defective child? 10%. You see? What I'm trying to say is non-disjunction happens in both genders. But because you only ovulate one egg as a female, if that egg is bad, is defective chromosomally, then the baby is going to be hurt by that, for sure, 100% sure. But in men, we got numbers on our side. So they worry more about the female age than the men. Don't misunderstand that. That means non-disjunction does not happen in men. No, it does. It does. It can.
What's the age?
Say?
What
We don't worry about the men. We worry about female age. Because of the chance event of happening in women, if it happens, the baby is for sure has a problem. So they don't worry about the age of men, for men. But what's the problem? The problem is non-disjunction. But non-disjunction can happen in meiosis 1, but it can only happen it can also happen in meiosis 2. Now look at the screen. On this side, it shows you the consequence to the haploids if the non-disjunction event happens in meiosis 1. Look, it is devastating. All 4 eggs or all 4 sperms are going to be bad. If the non-disjunction event happens in meiosis 2, which means meiosis 1 was fine. So this is a non-disjunction event in meiosis 2. The homologous chromosomes come next to each other, attach it, and make the tetrad. The homologous chromosomes separate in anaphase 1. They line up in metaphase 2. They go to separate. This one separates, but this one doesn't. That's a meiosis 2 non-disjunction. Meiosis 2 non-disjunction is more forgiving because half of them will be good, the other half will be bad. Now watch. The one with the N only, that's the good one because that's 23 chromosomes. The one with the N plus 1, that means 23 plus an extra one. That's not good. The N with minus 1, that means you actually have 22 because you're missing 1. It's 23 minus 1. Again, we're only talking about humans here. So that's not good. In this case, when the non-disjunction event happens in meiosis 2, at least 50% of the haploids are fine. The other 50% are defective. So let's say you're a female and you produce these haploids. I'm only going to worry about it when they're fertilized. Do you really think I worry about the menstruated egg? Nobody worries about that. I hope you don't worry about that one. The only one that you need to worry about is the one that becomes fertilized. You're worried about the baby having the problem, not the egg having it. If the egg has it and you menstruate it, okay, it's done, you're over, you got rid of it. It's the one that the sperm is coming in to fertilize. Now that one you're going to worry about because you just produced a zygote. So the concern is this. This scenario produces a condition called trisomy, means 3 chromosomes. The baby will end up having 3 of these. You look on the screen and say, where's the third one? I only see 2. Where's the third one coming from? I'm waiting for an answer. No. 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 Who said that? Fertilization. Who's that? You got it. It's coming with the sperm. If you're listening to me, are you going to worry about the egg that you're menstruating? Nobody is going to worry about that one because that's not going to develop as a baby. Yes, it's a bad egg, but you're getting rid of it through menstruation. The only one you're going to worry about is the one that is fertilized with the sperm who's coming in with a 3rd chromosome. So let's say this is chromosome 21. This baby, after the egg has been fertilized, will have 3 chromosome 21s. 2 from mom's non-disjunction and 1 from dad's sperm. He will be referred to as being trisomic. Say again? No, he will be Down syndrome. You're Down syndrome when you have an extra chromosome 21. You normally have 2 chromosome 21s and you're normal. When you have a third one, you'll have Down syndrome.
Only on that one?
On chromosome 21. Down syndrome is a trisomy of chromosome 21. Yep.
Can non-disjunction happen before the age of 35?
Oh yeah. Oh yeah. Non-disjunction can happen even in younger age, but what's the chance of it happening? A lot smaller. See, if I go buy a new car and I drive off with it, can it break down 3 lights after the dealership? Yep. Unfortunately. But what's the chance of a new car breaking down as soon as I buy it? Very little. You see? But can it break down? Yes.
Does it happen in mitosis?
No, this is non-disjunction of meiosis. We're talking about meiosis here, producing haploids. But see, the answer to your question, there is a chance anything can happen. But because the machinery is brand new, when you're younger, everything is still working. But then as you get older, things start slowing down, things start not working. So they figured out that around the age of 35, the chance of an event of one of these happening starts significantly going up. So that becomes the cutoff, the worry age. Now, does it happen? Does it mean when I'm 35, I will have a Down syndrome child? No. Just because my car has 150,000 miles on it doesn't mean it's broken. My last car had 220,000 miles on it by the time I got rid of it. And I had it for 17 years, and it was still going, and I cried when I let it go, right? But it was time to let it go. But just because you're old doesn't mean you're broken. But there's still a chance of it. So they worry about the female's age here. Now, in a non-disjunction of meiosis 2, it's more forgiving because in this case, when a sperm comes and fertilizes this egg, the baby is going to be normal. The baby will have 2 chromosome 21s, one from mom and one from dad. All right, so this one produces a trisomy event. 3 chromosome 21s, 2 from mom because of a non-disjunction, and 1 from dad. Sounds good? What about this one? This produces monosomy, means 1 chromosome 21. Because mom's egg does not have a chromosome 21 because of non-disjunction. Where did they go? They went over here. You see? Now when the sperm comes and fertilizes this one, the sperm is coming with a chromosome 21. So this zygote, will have 1 chromosome 21. That's not good either. So an extra chromosome 21 making you 3 chromosome 21s, that's trisomy. A missing chromosome 21, that's monosomy. Both of these is called aneuploidy. Aneuploidy is gamete, the number of chromosome are abnormal due to non-disjunction events. The fertilization of chromosomally abnormal gametes produced by a non-disjunction event. That's aneuploidy. And there are 2 possible outcomes, monosomy and trisomy. When you're N+1 as a haploid, that zygote is going to become trisomy. When you're N-1 as a haploid, the zygote will become monosomic. If you are N, then it'll be normal. Question?
So, what disorder will the monosomy have?
Oh, beautiful question. Hang in there, watch this. I'll tell you. So how do we test for this? So when the doctor says you should go get checked, you should go actually check your baby. The doctor is going to order what is called a karyotype. They literally go in with a very long needle, and they go into the amniotic fluid, the fluid where the baby is swimming, and they capture some of that fluid because they realize the baby is swimming in the fluid, so some of the baby cells are shedding into the fluid. So if I take some of the fluid, I can get some of the baby cells and do a karyotype on it. You literally take a picture of of the dividing cells to take a picture of the chromosomes and line them up. Okay, this baby has 2 chromosome 1s. Good. It's based on the length, the placement of the centromere, and the color pattern. That's chromosome 21. 2 of them. Chromosome 1. There's 2 of them. Chromosome 2. There's 2 of them. 2. Okay, everything is good. Everything is good. Everything is good. Uh-oh, hang in there. There are 3 of these. Once the doctor sees there are 3 chromosome 21s, that's a Down syndrome child. And she's female, because she's XX. Female. So you can determine gender. Now, you would not do a karyotype to determine gender. You would not do a karyotype at all unless you have in your mind that you will abort the child if he or she is Down syndrome. So the doctor is going to ask you first, what are your beliefs on abortion? No, it's against my religion, whatever. Then you don't do this test. You don't do the test just to find out if your baby is Down syndrome. You can just wait until the baby's born. You see? Because this procedure puts the baby at a risk of being miscarried. So I'm not going to put the baby at a risk of being miscarried just for me to find out the baby is Down syndrome and I do nothing about it. So usually people that have this in their mind, I will abort the child if the child is Down syndrome, then they will offer the test for you. But it all depends on your beliefs and religion and all of that stuff. Yeah. So that's a karyotype. So, the consequences. Or one other thing before I talk about the consequences. Can non-disjunction happen to the sex chromosomes? Of course, they're chromosomes. They can also do non-disjunction too. So there are some men that are actually XXY. What's the normal male? XY. They're called Klinefelters. The phenotype can range from no phenotype at all to extreme phenotype. It depends on the expression of the genes or whatever. But look at this one. Listen to this one. In normal males, they're XY, right? There's no bar bodies in normal males. And what's a bar body? X inactivation, right? Remember, if you have 2 X's, one of them is going to be inactivated, turned off. That's in females. Females have XX. They turn one of them off. So you can literally see it under the microscope. Oh, that's a bar body. That's a female. In this sample, if I get a sample from this male, if I don't know that the sample came from a male, I will look at the slide and I will say this sample came from a female because the sample has a bar body. The male is going to come up to me angry. That's my sample. How come you called me female? Well, I saw a bar body, that's why. I said, oh, that's your sample? Yeah. Oh, then I'm sorry, you're Klinefelters. Are we catching that? If that's a normal female, I'll see a bar body. If it's a male sample and he has a bar body, he's Klinefelters because he has an extra X chromosome that he will inactivate. Men, genetically normal males, do not have bar bodies. Genetically normal females have 1 bar body. Once a man gets an extra X chromosome, or sometimes maybe even extra 2 X chromosomes, then the bar body is, if I see 2 bar bodies in the man, then he is XXXY. You see that? Yes, sir.
What is bar body and how you spell it?
Bar body, B-A-R-R, body, B-O-D-Y. That's the X inactivation. Remember when we showed the cat with the fur color, the orange and the black? That's X inactivation.
Why does it matter if one's inactivated?
Huh?
Why does it matter if one's inactivated?
Well, what happens is which genes are on the X chromosome will determine the phenotype of the Klinefelters. If the genes don't do much, then the Klinefelters individual will show no phenotype. But if there are some genes on the X chromosome that are activated or inactivated that count for something, then you'll see in Klinefelters you can see a whole wide gradation of phenotype. Some Klinefelter patients get really no effect here other than them walking around with an extra X chromosome that's inactivated. Other Klinefelters have a drastic effect. Yes, sir.
Oh, that's the same question I was about to ask. Like, wouldn't it not have any effect because one of those would be.
Well, remember, the X inactivation is random. In some cells, this male will inactivate this one but turn this one on. In other cells, he will inactivate this one but turn this one on. Now it's a factor of which cell is doing what and what gene is on that X chromosome. You see?
Okay, but wouldn't he still have XY functioning properly?
That's right, that's fine. But again, it depends on which X chromosome is being active or inactive in which cell and what genes are on there that will determine the phenotype. Normally, you're just XY. So nothing happens with that. But now you're playing a game of which X, then you suffer the consequence of that. Yeah. Then you have females who have a missing X. Now, in this sample, if you give me a female that has Turner syndrome, and I don't know that this is a sample from a female, and I look at it and I don't see a bar body, I'm gonna call her male. You see? I don't see a bar body, male. And she's gonna come and yell at me, I am not a man, I'm a female. Well, I didn't see a bar body. I'm sorry, you're Turner syndrome. I don't even have to look at her slide. She will have classical characteristics of Turner syndrome. She will be sterile. She will have very delayed, if any, secondary sexual characteristics. Things like breast development, pubic hair in development, and so she will not be able to have babies. I mean, she's alive, at least.
So
Yeah.
about
Say?
the first century, the males,
Hmm.
would they be able to have babies?
Yeah, these are the whole gradation of phenotype, and it has to do with height and other things. Yeah. So I'm going to go back to your question from before. Listen carefully, please. Here's the summary of the aneuploidy effects. Aneuploidy is either a missing chromosome or an extra chromosome. An extra chromosome, we called it trisomic. A missing chromosome, we called it monosomic. Good. Listen carefully. The only monosomy, you asked this question, which is a beautiful question. The only monosomy that survives is this one. What if I have missing chromosome 1? You're miscarried. What about missing chromosome 10? Miscarried. 5? Miscarried. Think about it. Chromosome 1 has thousands of genes on there. And a lot of them are involved in your development as a baby. And it goes missing on you. Oh, you're in trouble. Why? Because you can't develop as a baby. You get miscarried. So there are women that have, hey, I'm pregnant, and then 3, 4, 5 weeks later, I had a miscarriage. It's probably because of a chromosomal defect. That's why. The baby had a chromosomal problem and was miscarried. Not allowed to progress. Sorry. Can't go through life without a chromosome 10. I'm sorry. But that's the only one that survives. But the consequences of it right here. So monosomy X is the only one that survives. All the other monosomies die, miscarry. Okay, trisomy. Listen carefully to the words here. Only trisomy 21 reaches adulthood. That's it. That's Down syndrome. Trisomy 21, which is Down syndrome, is the one that is allowed, the baby is allowed to be born and develop and grow and lead a somewhat functional life. Now, of course, there are consequences, of course, to Down syndrome. But at least survives, at least reaches adulthood. All the other trisomies die. But here's an interesting one. There are a couple of trisomies, can't remember the exact numbers of them, but there are a couple of chromosomes. They would allow you to be born live for a year or so, and then die. And it's a miserable year of life. You wish you were never born. So they're devastating. So you would think having an extra chromosome would make me smarter, would make me taller, would make me faster. No, you're sick. Because too much of something is not good. Do you remember me saying that before? I told you before in biology, too little of something is not good, too much of something is not good. What's good? The right amount. And the right amount is 2 chromosomes. Do you see that? For each chromosome. So all the trisomies eventually end up dying except for Down syndrome. That's the only trisomy that reaches adulthood. And if you notice something, why? It's because it's the smallest chromosome. It is an extra piece of chromosome, but it's the smallest one with the lowest number of genes, most likely, and that's why it's allowed to survive. But it's still devastating as a disease. Yes, sir.
Considering you said 21 was the smallest one, if the person had trisomy like on the first one, which is the biggest one, how long would the child, the pregnancy even last?
I don't know that. They don't. I mean, it's miscarried. No doctor will find out why you miscarry. You see what I mean? Unless you repeatedly miscarry, then they have to figure out, is it hormonal, is it whatever? Then you work with the patient. But for you, if you miscarry, they're not gonna try to capture it and test it genetically and all that. You miscarried. Good luck next time. That's all. So some students sit here in the room and say, "Whew, thank God I don't have an extra chromosome 21." Well, anyways, you wouldn't be here, right? "Whew, thank God I don't have a missing chromosome." But you look at this. You don't have to have an extra chromosome 21 or whatever, or an extra chromosome this or missing that even a piece of chromosome is a problem. What if you go missing this piece of the chromosome? Oh, that's a big problem for you. When does that happen? During DNA replication. We'll talk about it in the next chapter, most likely on Monday. During DNA replication, if the enzyme decides for whatever reason, I'm not going to copy D, oh, you're in trouble. Let me give you an example. What if D is a checkpoint guard? Oh, you mean you want me to remember all the other stuff to understand this question? Yeah. What is D? What if D is a checkpoint guard? Which disease will you have? Cancer. Cancer. If D goes missing, that cell is going to become a cancer, or a tumor at least. Now you know the importance of that. Your chromosome has A, B, C, D, E, F, G, H. When it goes copies itself during DNA replication, you need all of them. If your enzyme by mistake forgets to copy D, then the new chromosome is going to look like this. That's called a deletion. A deletion of a checkpoint guard? Oh, you're in trouble. That cell is in trouble. It will go through the cell cycle unchecked, and it will become a tumor, potentially a cancer. Yes, sir.
Wait, wouldn't be a deletion of D cause a ripple effect to the rest of your gene to the point where that protein that make up that DNA will be completely ineffective for DNA?
Only this cell will be defective,
Oh,
because the same event happening in the next door cell may be fine. This particular, see, that's what cancer starts as, one cell making a mistake. Then that cell starts dividing out of control to produce the tumor. One common mistake that we find in cancer cells is this one, deletion. Pieces of the chromosome go missing on you. That's why it becomes a cancer. Here's another possibility. Some cancer cells are produced as a result of a duplication event. The same enzyme that forgot to do this one did this one twice. Look at that. It copied BC and then it did it again. You got BC and BC. You think you're taller, smarter, and faster. No, you have cancer.
Wow.
You see that? just a duplication of a piece of chromosome. And now it all depends on which piece and whatever, and how big it is, and all, but that we've seen in cancer too. When you isolate a cancer out of somebody, when you take cancer out and then analyze why did it develop into a cancer, you're gonna see that some of the cells in that cancer became cancer because of a deletion. Other cells became cancer because of a duplication. I'll get to your question. Other cells became cancer because of an inversion. The order of the genes is supposed to be A, B, C, D, E. It ends up being A, D, C, B, E. It flipped the order of the genes. That's not good either. We've seen cancers with these mistakes too. Then there's a translocation. A piece of chromosome 10 goes to 3 and a piece of 3 goes to 10. That's like me telling Kai and Devin, exchange seats. But Kai always sits here and Devin always sits there. Well, I'm gonna, that's not good either. That's called translocation. There are some cancers. We've seen all of them in one type of cancer. One type of cancer will have all of them. They will also have the extra chromosomes and the deleted chromosomes on top of that. When you look at cancer cells, you realize something. Anything can happen. And that's why that's the reason why they're cancer. You see? Go ahead, somebody had a question. Go ahead. Go ahead.
I'm wondering why, let's say I have a lung cancer. How does your body know, say, it stopped growing at this specific point? Let's say, why don't my lung go to a different shape? Takes a different shape.
I don't know about that. I'll be honest with you. But come and talk to me after that class and see if I can figure out what you're asking there, okay? So here are some example diseases. One example from a deletion. Chromosome 5 deletion causes, obviously it's a specific deletion of chromosome 5, causes a disease called, if I can pronounce it right, Karyotyping. That's French. That's the best I can do, all right? Which translates to the cry of the cat. Why is that? Why is it called that? Because babies or little children that have this deletion, when they go cry, they literally sound like a cat for a little while, until they get a little older, then they're fine. But they still have the disease, of course, but they're fine. But it's identified through the cry of a baby, and when you listen to them, they literally sound like a cat, because the disease has an effect on the vocal cords. So it makes them sound like that. But as they get a little older, they overcome that. Their vocal cords change, of course, with age, like we all have our vocal cords change with age, with hormones and stuff. And they do too, and then they lose that effect. And I just mentioned that things like deletions, duplications, inversions, translocations can cause cancer. So they're showing you an example of a leukemia, cancer of the white blood cells, caused by a translocation event. Just some examples that are discussed in your textbook. Okay, I think I have your attention so far, and I love that about you guys, but I need more attention now. This one is tough. Genomic imprinting is the next topic. It's a tough one for some students. I'm gonna try my best explaining it to you, and I have a paper here of a workup of what I'm gonna show you on the board so I don't get it wrong. Okay, so please focus with me, please. Some traits, so this is genomic imprinting. In genomic imprinting, the genes, or the way they work, they're very interesting here. Now, not all your genes are gonna do this. Genomic imprinting is dealing with, for example, a handful of genes. Let's say about 70 to 75 genes do genomic imprinting. Out of your thousands and thousands of genes, only a handful of them do this, called genomic imprinting. So we're talking about things that happen with your genes that are out of the ordinary. You can't do dominant recessive and all that. They behave really weird. Genomic imprinting is all about gene silencing. You're causing one allele to silence while keeping the other one on. That's genomic imprinting. How do you silence a gene? You methylate it. If you remember back in one of the chapters, 4, when we talked about the functional groups, one of the functional groups was a methyl group, CH3. I kind of quickly mentioned it back then, but here's the formal discussion on it. You'll see in the next chapter or two, your gene or genes are arranged this way. This is your gene, and there's a control switch called the promoter. The promoter turns this gene on. Or it can say, no, we don't want it on. One way to control the promoter is to either methylate it or not. So watch this. If you methylate the gene, the promoter and the gene, if you add CH3 groups to the gene and the promoter, you turn it off. Genes are in the off position when they're methylated. That means when you remove the CH3, what happens? You turn it on. So a gene that looks like this, without the methyl groups, would be on. With the methyl groups, it's off. Without the methyl group, it's on. So I can control when do I turn off the gene and when do I turn on the gene. It's amazing. Your cells do this. It's called methylation. Methylation. Through methylation, I can determine whether the gene should stay on or off. Are we understanding that? And that's the control of gene expression. So you have to understand that before we go to the next step. The next step is this. So now we know how we quiet or silence genes. We methylate them. Good. So where does genomic imprinting play a role here? Well, let's see. This is your dad, for example. Got 4 genes, gene A, B, C, and D. Well, mom is no different. Mom has the same 4 genes, but they're lowercase letters. Are we catching it so far? Obviously, you are a product of mom and dad. Your dad's sperm fertilized your mom's egg, and that's how you came about. So you are this. Who did you get this from? Who did you get this from? Yeah. Everything good? I'm just throwing random letters on here. Now here's the interesting thing. These are 4 out of those 75 genes that I was talking about. About 75 genes or so will do this. In dad's sperm, keep A on, turn B off. Keep C on, turn D off. How do you turn it off? Methylation. Are we okay now? In mom's egg, whatever dad did, I'm gonna do the opposite. If dad kept this one on, I'm gonna turn it off. If dad keeps this one off, I'm gonna turn it on. You catch that? So dad keeps A and C on, mom keeps B and D on. And the ones that are starred are the methylated ones. So the starred ones are the methylated. We can also call them the genomically imprinted ones. They're the ones that are off. The ones that are not starred are the ones that are on. That's dad's sperm doing that. And that's mom's eggs doing that. But then here's you. You're the zygote. Which A are you gonna express? Dad's or mom's? Dad's, because mom's is off. Do you see that? Which B is gonna affect you? Dad's or mom's? Because dad's is off. Which C is gonna affect you? Because mom's is imprinted or methylated. What about D? Dad is off. Yeah. So if you look at this, the expression pattern of the genes, whatever the A phenotype is, you're getting that from dad. Whatever the A phenotype is, A phenotype for you will be similar to your dad's, because your A from mom will always be off throughout your whole life. That means you will be like dad for A and C. Am I right? The C phenotype, you will be like dad. But what about B and D? You're going to be more like what? You're going to be like mom. One of those genes is the insulin growth factor gene. The insulin growth factor plays a role in your growth while you're a baby, developing as a baby, because I told you that all the genes that are involved in genomic imprinting are involved in your development. I may not have said that actually, but I'm saying it. All the genes involved in genomic imprinting are part of your development in utero. One of them is IGF-2, which is insulin growth factor 2. Which insulin growth factor 2 gene are you going to be expressing? You're going to always be expressing dad's. Everybody in this room, I don't care what gender you are expressing dad's IGF-2, not mom's. Because the female IGF-2, the mom's IGF-2, is always going to be imprinted. IGF-2, at least in some animals, determines the dwarfism phenotype. Some mice, for example, are dwarfs because of a defective IGF-2. They're not going to blame mom for that. They're going to blame who? Dad. Because it's dad's IGF-2 that will determine your IGF-2 expression, because mom's IGF-2 is always off. So A and C phenotype will come from your dad. B and D will always come from your mom, because the other counterpart is genomically imprinted. If you thought that was tough, now listen to the rest of it. So this is you sitting in here today. Your dad's A allele is being expressed in all of your cells. Your mom's A allele is turned off, regardless of what it is, because it's been genomically imprinted in the egg that produced you. Fast forward until you get to puberty. Then what happens? Hang in there, watch this. all of your somatic cells will still do this. Your skin cells, your liver cells, your heart cells, that's you for the rest of your life. But if you are a female versus a male, the male is going to produce sperm. The female, obviously, will be producing eggs. And they're going to be haploid. So let's say you're a male and you have this. What haploids are you going to produce? Watch this. Watch this. The male is going to produce 50% this and 50% that. Do you agree?
Yes.
Okay, let's do this here. The male sperm will have A, B, C, D. And the other one, A, B, C, D. Now catch this one. When you, as a male, develop the sperm, this one, because it came from dad, will stay the same. A is active, B is off, C is active, D is off. But watch what you're gonna do with this one. You're gonna A, B, C, D. You're gonna remove the methylation and remethylate it at B and D. Did you catch that? When you go and produce haploids, the haploids that you produce will be based on your gender. Even though you are expressing A here, turning this off here in your somatic cell, the male sperm will have the B and D always turned off. And it doesn't matter which chromosome you're talking about, dad's or mom's, because this is dad's, this is this one, and that's this one. You're literally gonna remove the methyl group from A and put it on the B, remove it from C and put it on the D in your haploids. So the men in the room will always give the active A and the inactive B, the active C and the inactive D, regardless of which chromosome they originally got it from. Well, the same thing for the female. The female is going to do the same thing, but she's producing eggs. A, B, C, D. A, B, C, D. She's female. So she's going to do the female imprint. The female imprint is A off, C off. Well, that's this one anyways. This one, I gotta remove this one, put it here. Remove this one, put it here. Because I have to have the A and the C off because I'm female. It's fascinating. It's not only fascinating that this happens, it's fascinating that we figured it out. Do you agree with me? I mean, there are people that actually worked on this and figured this out. I mean, we have a hard time understanding it. They actually figured it out in research, through research. You are a byproduct of your dad and mom's fertilization event. Dad will always, the male will always have A and C on, B and D off in the haploids. The female will do the other pattern, A and C off, B and D on. Then they fertilize, that's you. The A phenotype is from your dad. The C phenotype is also from your dad. But B and D is from your mom. That's your somatic cells. But once you start producing haploids, listen to me, you're gonna erase the the imprint and re-imprint. Erase the imprint and re-imprint based on your gender. The males will always have B and D off or methylated or imprinted. The females will have A and C. Now, obviously, I told you this These are just 4 genes as an example on the board. We're talking about 75 of them will do this. And they're all involved in what? In your development. So luckily we have it videotaped now, and now you can follow through at home. If you rewatch it at home, hopefully it'll help you there. Okay, the last thing I'm going to talk about and send you on your way here is another weird way of inheriting genes. You probably already kind of like know about a little bit of it because I did hint to it before. It's the genes in your mitochondria. You remember, where do you get the mitochondria from? Where did we get the mitochondria from? Bacteria. But for you, you actually get it from your mom, right? So bacteria originally, but you get it mostly from your mom. So watch this. Mom gives you 23 chromosomes on the egg. Inside the egg, you have 23 chromosomes, and there's a lot of mitochondria in here, in the cytoplasm. And each mitochondria has circular DNA called mitochondrial DNA. Of course, they have genes. The mitochondrial DNA have genes. A lot of the genes on the mitochondrial DNA are involved in cellular respiration because the organelle is involved in cellular respiration. All right, here comes Dad. Dad's sperm, 23 chromosomes, maybe a few mitochondria. Obviously, because the cytoplasm is a lot smaller, so he'll have fewer mitochondria. The main purpose of this sperm here is to deliver the 23 chromosomes. So I tried to make a joke about that. Dad gives you 23 chromosomes and leaves. You don't see him until you're 18 or whatever, maybe. Anyways, but he actually gives you some mitochondria, just a few of them. So some of the mitochondria that you have may be coming from Dad, if they're still persisting there. But where did you get most of your mitochondria from? From who?
Mom.
From Mom. There are genes in there, and they will carry, there's inheritance, there's genetic problems that can happen as a result of you inheriting a defective mitochondrial gene and so forth. But you would probably more often than not blame that on Mom. So more often than not, you would blame a mitochondrial inheritance problem on your mom because Mom is the one that gave you most of your mitochondria. So that's obviously a special type of inheritance there, mitochondrial genes. You guys good?
Yeah.
A lot of information? You know this information goes nowhere unless you do something with it. You agree with me? About half of the information I talked about today will evaporate by the time you get to your car. The other half by the time you get home. You need to do something with the information, you guys. I'll see you on Monday. Watch the videos, please. Watch the videos.